Today we're talking about exactly how scientists have been using virtual reality setups to study lab mice. But before we get into that, in a landmark decision, the FDA greenlit 2 new drugs for the treatment of sickle cell disease in patients 12 and older, one of which, Vertex's drug Casgevy, is the first approved use of genome editing technology, CRISPR, in the U.S. Bluebird Bio's Lyfgenia also is a cell-based gene therapy. However, it uses a different gene modification technique to deliver tweaked stem cells to the patient. Both approvals cultivate new pathways for the treatment of sickle cell disease, which is an inherited blood disorder that is characterized by red blood cells that can't properly carry oxygen, which leads to painful vaso-occlusive crises, VOCs, and organ damage. The disease is particularly common among African Americans and, to a lesser extent, among Hispanic Americans. Bone marrow transplants are currently the only cure for sickle cell disease, but they require well-matched donors and often involve complications. The FDA's decision comes shortly after UK regulators, as well as the National Health Regulatory Authority in Bahrain, both approved Vertex's Casgevy. The approval for a CRISPR-based treatment creates opportunity for further innovation in the gene editing space for treatments ranging from cancers to heart diseases to Alzheimer's. Gene therapy holds the promise of delivering more targeted and effective treatments, especially for individuals with rare diseases where the current treatment options are limited. Nicole Verdun, director of the Office of Therapeutic Products at the FDA Center for Biologics Evaluation and Research, said, Casgevy is still currently under review by the European Medicines Agency. Believe it or not, scientists have been using virtual reality setups to study brain activity in lab mice for years. In the past, this has been done by surrounding the mice with flat displays, a tactic that has obvious limitations for simulating a realistic environment. Now, in an attempt to create a more immersive experience, a team at Northwestern University actually developed tiny VR goggles that fit over a mouse's face and most of its body. This has allowed them to simulate overhead threats for the first time and map the mice's brain activity all the while. The system, dubbed Miniature Rodent Stereo Illumination VR, or iMRSIV, isn't strapped onto the mouse's head like a VR headset for humans. Instead, the goggles are positioned at the front of a treadmill, surrounding the mouse's entire field of view as it runs in place. We designed and built a custom holder for the goggles, said John Issa, the study's co-first author. The whole optical display, the screens and the lenses, go all the way around the mouse. In their tests, the researchers say the mice appeared to take to the new VR environment more quickly than they did with the past setups. To recreate the presence of overhead threats, like birds swooping in for a meal, the team projected expanding dark spots at the tops of the displays. The way they react to threats like this is not a learned behavior, it's an imprinted behavior, said co-first author Dom Pink. It's wired inside the mouse's brain. With this method, the researchers were able to record both the mice's outward physical responses, like freezing in place or speeding up, and their neural activity. In the future, they may flip the scenario and let the mice act as predators to see what goes on as they hunt insects. A paper on the technique was published in the journal Neuron on Friday. Thanks for listening to the show. Make sure to rate, review, and subscribe on Apple Podcasts. Today's show featured journalism by Engadget contributors Malak Saleh and Cheyenne McDonald and was produced by Spoken Layer. I'm Imran Sheikh, and we'll talk more tomorrow.
Spoken Layer.